I'm Madhu Anand-Srivastava. I'm professor at the University of Montreal, Department of Physiology, Montreal, Quebec. We have been working on guanine nucleotide binding proteins, also known as G proteins, which play an important role in the regulation of a variety of physiological functions, including vascular reactivity and vascular tone. What we have shown is that the expression of GI regulatory protein, which mediates the inhibitory responses of hormones and inhibit adenylyte cyclase activity, decrease cyclic AMP levels, are increased in various models of hypertensive rats, like genetic model of hypertension, that is also known as SHR, spontaneously hypertensive rats, Doka-Salt hypertensive rats, and some other hypertensive rats. And this increased expression is of GI regulatory protein is the causative factor for the pathogenesis of hypertension, because the inactivation of GI regulatory protein in the very early stage of pre-hypertensive rats attenuated the development of blood pressure and suggested to us that enhanced expression of GI regulatory protein may be important contributing factor for the pathogenesis of hypertension. And the second work which we are doing is atrial natriuretic peptide receptor C, which has been shown previously that it has a clearance receptor. It is a clearance receptor and does not have any physiological role, but we have shown that it is not a clearance receptor only, but has physiological functions. So we have also shown that CNP, which is a ligand, specific ligand for NPRC receptor, attenuates the enhanced expression of GI proteins in spontaneously hypertensive rats. It decreases the oxidative stress also. So we were interested to see whether the in vivo treatment of CNP could attenuate the development of high blood pressure. So we treated the SHR with CNP for about 8 weeks and at different doses, and we have shown that CNP treatment of SHR decreased the attenuation of blood pressure, and we were interested to see what could be the mechanism. So we have shown that CNP decreases the enhanced expression of GI proteins, decreases the oxidative stress, decreases the nitric oxide level, decreases the nitrooxidative stress, and but did not have any effect on the levels of NPRA or NPRB receptors. Thus, we have shown that CNP, which was shown as a silent receptor and did not have physiological role, we have shown that this receptor is physiologically active, and activation of this receptor attenuates the development of blood pressure, and we suggested that CNP, a ligand of NPRC, can be used as a therapeutic agent for the treatment of cardiovascular diseases, including hypertension. I think it's very important because there are so many aspects of cardiovascular disease which are discussed here in this meeting, the basic science as well as the clinical science. And I feel that it's a very interesting conference and will be helpful for various people in the field.